So, hi everyone. I'm Hazel. Welcome to the Single Cell and Spatial Club today. And as you can see on the screen right now, today we are excited to introduce the latest evolution of Talk2Data. And because this is all new, so please let me know if you have any question or anything you want to know more along the way. Feel free to put it into our chat section, and we will try to answer all of your questions. Okay, so today the main reason why we're here is to introduce you. So what is new within the Talk to Data? Okay, so now let's go directly into the new stuff of our platform. So the first thing I want to share with you, and also one of the most significant updates, is our expanded database. We've broadened our scope to include both, as you can see, both single cell and bulk analytic data. The spatial data is also on the way, and it will be available here very soon. And this makes the platform more versatile. And at the same time, we also added more species like human, mouse, and non-primate, um, giving you access to wider range of data in your research. And also on the right side of the screen here is one of the most, one of the highlights of our different data. So let me show you this table for more, clear information of what databases that we have. So this table here highlights all the new databases that you now can explore. You can see that the AI Sonya 1.0 is they are actually our curated databases for human, mouse, non-human primate, and also single cell and bulk analytic. However, besides that, we now include curated datasets like the human cell atlas and the Tahoe 100 million. And this consolidation allows for more comprehensive analysis and discovery. And there are a little star here. So actually, this is a very interesting news to share with you guys that right now, the Tahoe 100 million and the Human Cell Atlas they are open for public to explore. So later on in this presentation, I will share with you so how you can explore these databases using Talk to Data without the need of of a license from BioTouring. Okay, so again, I just want to share that now we have in total of like around 500 million cells to explore. Uh, First of all, we have 200 million cells from human. We then have another 60 million cells from mouse and 6 millions from non-human primate. Okay, so this is one thing that we really proud of because of the size of the database that we have. Okay, and also within this new Talk to Data, we now bring AI to Talk to Data. And here we're introducing Sonya, our new AI agent. And Sonya has been trained on extensive biomedical data to help you with your research. So right now, if you go to Talk to Data, you'll be able to see Sonya right here on the right side, I'm sorry, on the left side of the screen, where you can just open her and ask her any biological question that you have. For example, here, I already asked her some questions like here, like what are the marker genes of T cells in lung cancer? And Sonia will provide like a very concise data back answer, and it will save much of your times in your analysis because right now you just need to ask. And also, what more advanced about Sonia is the fact that for each biological keywords in her answer, you can actually track back to what data set that we have in our database that related to this answer. So it's like more layers to confirm the findings. Okay, and then another thing, another thing for this is actually for more of our technical users. Like we introducing the BioFlex SDK. So okay, let's understand these SDKs as a powerful toolkit that allows you to directly integrate Talk to Data's capability into your own custom pipelines. And what does it mean? Basically, as you can see, here's a lot of code, and that means you can programmatically access the data Like you can find the data, run analysis, and leverage our platform's power without leaving your own workflow. You can run the code. You can just install the SDK and query the database. Okay, and also yes, the BioFlex is one of the options that you can see when you open Talk2Data. Okay, so now we go to the actual the actual ability of Talk2Data in terms of data explorations. Here, um, here we also introduce a new way to extract and explore individual study within our platform. So here, this is the new interface. This is a new interface where we hope that it will make it much easier to find the exact study that you need. So on the left side of the screen, that you can see that you can now search and filter studies using like a wide range of standardized criteria from specific cell types to different treatment conditions. So this new functionality helps you to quickly narrow down the results and focus on the data that matters most to your research. Okay, so that's the filter panel on the left. Now we go to the central panel. So after you filter your studies, you can then quickly review the metadata because we understand that even after filtration, there are still lots of information that included in each of the data set. For example, here we search for like breast cancer studies that having all kinds of treatment response. So after you filter, you can go to the central panel to actually view. So in individual study here, what type of treatment were used and what type of response were there. And so that that this allows you to make like informed decisions without having to open each study individually. And because as you may know that opening each study can take time. So now we try to save the time by showing you all the information needed right here. And not only this one, not only this overview, we also have the like um like a comprehensive view of the data where you can see all the key information ranging from the actual abstract from the paper to the UMAP showing the cell population of that specific study and also the metadata included, all metadata included. And again, this we hope giving you a complete picture of the study before you dive into the actual data. And and after you find the right study, you can open it in Browser Pro. And this give you like a more in-depth and also as you can see here, a more interactive view of the data. And in Browser Pro, you can perform more detailed analysis, visualize specific gene expression, and then explore the dataset in full detail. And here, this is one of the many features that you can perform on Browser Pro. For example, the cell-cell communication. And later on in this presentation, we will show you a quick, use case to maybe help you get a little bit more understanding of how you can utilize different features in Browser Pro and then integrate that with the whole database of Talk2Data. Okay, so that's like for single study exploration, and now it's actually the real highlight of this update. So this is our new workflow. We introduced a new workflow for cross-database exploration. We hope that it will reduce your clicking time, it will reduce your searching time, and it will show you more information on one single screen. Okay, so this is the new interface. This is the new interface where we show you, you just need to put in the gene and we will show you all the information that you needed, like what cell types express the gene. You can also filter for what disease, in which disease. And after you like query for any specific genes, you can even learn more about that genes, like know more about, so what is this gene about? For example, we have some TH here. representing for the dopaminergic neuron and showing like not only the information about the gene, but we also find you like the co-expressed gene. And in here, let me go to the dopaminergic neuron, for example. If you click into any of the cell types within this interface, you will be able to see the top 100 marker genes right away. And I just want to spend like let's say a few seconds to tell you that this is a huge computational strength from our team. So in here, we are talking about 200 million cells. And in real time, we calculate the marker genes for any cell type in like, across the 200 million cells. As you can see here, when you select the dopaminergic neuron, we will find the genes that are specifically expressed in dopaminergic neuron versus the other cell types in the brain tissues. And we calculate it real time in just 2 seconds. And that is very impressive for the team to make it happen. Okay. And what else that you can do? What else you can do here? Of course, you can run the DGE. You can run the DGE as well. And the DGE, we hope that we also make it more streamlined for you to explore, where the fact that the fact that you can just select the differential gene expression and like you can directly select the cells group that you want to work with from the composition tree. This is, this is one attempt of us to reduce the clicking time. You can go directly from the composition to finding the differentially expression genes. So this is one example of finding the differentially expressed gene between the normal and PD dominant neuron. And again, we calculate it in real time within 2 seconds. Okay. This is DGE. And like after finding the DGE, what you can do further is to find the co-expressed genes. And in here, if you click on any of the gene name, you can find the co-expressed gene in like, in a specific biological context. Okay, so that's like almost everything that you can do with the current Talk2Data, like I'm sorry, the new Talk2Data. And so, okay. I'm sorry. And after this, we will show you like Talk2Data interaction. So before we go into our little use case, I just want to share again that even if you're not a biotrain users, please feel free to explore Talk2Data using the talk2data.biotrain.com. And when you open talk2data.biotrain.com, what can you explore? You will be able to use all the features within Talk2Data that I just shared with you. And you can, I'm sorry, you will be able to use all the features that I just shared with you using the public databases like the Human Cell Atlas and the Tahoe R Million. This is an example of the Human Cell Atlas. And what further that you can do is also to explore individual study. So that means you can not only exploring Talk2Data, but you can also explore V projects as well. And just, sign in and explore it yourself. And so from here, I want to share one example workflow in like investigating the cell-cell communication in lung disease and, figure out how we can validate the finding using the Talk2Data database. So how does it start? First of all, let me go to the actual Talk2Data. Let me go to Talk2Data. Now we will go to the studies tab here. So in the studies tab, you can find all the study. And I want to go to the Human Cell Atlas, right? In the Human Cell Atlas, let's look for the, wait a second. Let's look for the lung disease. Okay, let's look for the lung disease. Okay. And this is the integrated cell atlas of the lungs in health and disease. We can explore it right away. Okay. And after we explore it right here, so as you can see, I already opened it. So after we open it, I want to investigate. So actually, we already investigated that. So in this lung atlas, we have 16 different types of diseases. let me scroll here and I can show you the different condition of this here, different condition. Oh, should not be there. Okay, the condition here. There are 16 different conditions ranging from COVID-19, like different, pneumonia to different kind of fibrosis and different kind of lung cancer. And we want to focus in the lung adenocarcinoma and the squamous cell adenocarcinoma, basically lung cancer. And we want to find out, is there any pair of ligand receptors that we can found? And we already run that using the cell-cell communication right here. And let me go back to the slide where I can show you. So what do we find? Okay, so using different cell types, metadata, and split it by different conditions, we have found a very, let's say, significant interaction. So using the cell-cell communication, we found that within the bronchus fibroblast and the type 2 pneumocyte in different kind of lung cancers, we always see the high expression, or let's say the high interaction between the FN1 and the SDC1, in which FN1 is the ligand and the SDC1 is the receptor. And in here, I also share with you a quick information about what is FN1 and what is SCD1. I'm sorry, SDC1. The FN1, or the fibronectin 1, is a major component of the ECM. And these cells, these genes express a lot in cancer-associated fibroblasts and may initiate the metastasis, may initiate and control the metastasis of the tumor cells. And then, so of course, that's why we have the fibroblasts here. And then for the receptor, the SDC1, or the syndicant-1 is associated with poor prognosis and the epithelial mesenchymal transition, or the EMT, and again is a property of metastasis. And we, based on the literature review, we also found that the FN1 and the SDC1, when combined together, can, control and push the metastasis to go forward. So yes, that's why we found a high expression of FN1 and SDC1. So after we find this, this interaction in different kind of lung cancer, we also ask another question. So whether this FN1 and SDC1 interaction, does it occur in all type of different lung cancer data, or it only occurs in this specific atlas of the human cell atlas? So in order to answer that question, we go to the bioturing curated database of 200 million cells to try to validate this finding. Okay, so how did we validate that? In here, you can see that we filter for lung tissues of, two cell types that we just saw earlier, the fibroblast and the pneumocell, and we query for the expression of FN1 and HDC1. Okay, let me show you in action. Okay, let me go back to the Talk2Data here. Now we go to the analysis. Not human cell atlas anymore, because we know that the interaction is there, highly expressed in lung cancer of the data from human cell atlas. Now we switch to the bioturing database, and in the anatomy, I want to filter for the lung. I apply it. In the cell type, I want to filter for the, here I want to have the fibroblast of lung and apply. And I also want to filter for the type 2 pneumocyte. Okay, type 2 pneumocyte. Okay. Now FN1 and SDC1. I already query for it. So now I can open here to view. And this is, as you can imagine, this is one composition tree of level 1 is the lung or the tissues. Level 2 is the cell type. Now that I want to compare the two genes expression in different conditions. So what can I do? I can include another level of conditions in this composition tree. So in order to do that, so I think that this is a good tip to keep in mind. So this button right here, the custom tree view, is the like an important feature to remember. Okay, we can, here we have the tissue, the cell type, and now we go to the conditions. I update it, and now you can go and you can see the different expression of SM1 and SDC1 in different type of diseases in the lung tissues, coming from, from type 2 pneumocyte or the fibroblast of the lung. Okay, uh, let me sort, and let me also sort this one and sort this one first. Let me sort this one first. And you can see right away. It's very interesting. It's very interesting that again in the Talk2Data database, we see the high co-expression of FN1 and SDC1 in the lung cancer. Here, squamous cell carcinoma and squamous cell lung carcinoma. But we don't see that, that much of co-expression in other type of diseases. Okay, we don't see that, as you can see here. And if you may remember, back to the slide here, the FN1 is the ligand and is expressed in the fibroblast, higher in the fibroblast. In here is the same. And when we go back here, we see that the SDC1 is a receptor. It's highly expressed in pneumocyte. Now we go back and let me scroll all the way up. Okay. And you can see the same situation. We have squamous cell carcinoma and squamous cell carcinoma of high expression SCD1 in type 2 pneumocyte. So that's like a way of us to try to, validate what we found in the human cell atlas. And so we can say that we found high expression of SN1, SCD1 in squamous cell lung carcinoma and also type 2 pneumocyte in our bigger database. Okay. And so from this point, so this is after validation. And what else you can do with Talk2Data from this use case? Okay. So now we have SN1, FN1, and SDC1. So what if, because let's say that you work with the drug discovery or you want to find the biomarkers. So this is a good point to actually go and find more target, find more candidate so that you can investigate that further. Okay. So let's say that for FN1, it is highly expressed in the in the fibroblast of the lung. And let me just deselect the type 2 pneumocyte for a moment. So I can show you that when you hover to the FN1 gene name and click on this view information button, okay, we will show you directly the list of co-expressed gene with FN1 in fibroblast of the lung. And these like the CO683, the CO182, these can be other target, other candidate. Maybe it co-express with a gene that moderate the metastasis. So maybe they can have like maybe similar role, or maybe we can attack different multiple target at a time. Okay, so that's what you can do with the FN1. You can do the same with the SCD1, with the SDC1. You can just view the information and it show you the co-expressed genes again. Okay, and and from this point here, another thing you can do, so this is like 10 other different targets. So what else you can do? The other things that you can do is to find like a broader understanding of, of the either lung carcinoma and other disease. You can run the DGE as well. So DGE is one option up here. Just click on the DGE and you can either select the group, like this can be group 1, and you can select the normal as the group 2. I'm sorry, as the group 2, the normal as the group 2, and oh, I'm sorry, I should go back to group 1. And okay, This is normal, and this is the fibroblast of the lung, group 2. And we can run the analysis that way. As you can see, it's very straightforward. You don't need to think much about what should we do, what button should we click. We just need to select and run the analysis. And now that we have the list of genes, we have another target to work with. And yes, that is like an example use case of what you can actually do to investigate further on Talk2Data. We hope that even if you are a Byjuan user or you are not a Byjuan user, feel free to explore this database because this is very precious to work with. And let us know if you have any feedback, anything that you think that may have us to make it better, or any input you may have, and we are happy to have. So yes, that's also the end of my presentation for the new Talk2Data. And here, here we are showing you on the screen is a little, little survey for, some feedback on Talk2Data so that we can develop it better. Like, please let me know along the side, along the way. I have shown you a lot, like different ways to study, the analysis features, the AI, the Sonya, the SDK. Please let us know which features are you most excited to use, and what else that you would want to have on Talk2Data. Please let us know, and we are happy to, help you with that. And we're happy to discuss on that. Okay, so I will open the poll for another 1 to 2 minutes, and then I will let's see whether we have any questions, and we can end the sections right here. And yes, thank you a lot for your attention and your time being here. Yes. Yes, okay. So that's it. Thank you so much for being here today. We will post this video again, this section again on our YouTube. So please feel free to share and to watch again. Yes, thank. Oh, I'm sorry. See you next week.